Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is plant development. This video is taken from Plant Physiology series CBSE class 11th topics, part 2nd of chapter 16. Now, let's see here the lecture overview. So in this video, we will discuss on differentiation, de-differentiation, redifferentiation, development of plants, difference between growth and development. So let's start the topic. Here our first topic is differentiation. So what is differentiation? The biochemical or morphological change of meristematic daughter cells to become a permanent cell is called differentiation. During typical meristematic growth, a meristematic cell divides by mitotic divisions to form two daughter cells. Out of the two cells, one may retain the meristematic activity and prepares biochemically to divide further. The other one may elongate or taken up any other morphological shape depending upon the requirement of the organ where growth is taking place. For example, a cell situated at the dermal layer of the root apex may elongate to form a root hair, or a cell situated centrally may elongate to become a cell of xylem or phloem. Now, de-differentiation. So, the phenomena of regeneration of permanent tissue to become meristematic is called de-differentiation. So de-differentiation is an important biological phenomenon whereby cells regress from a specialized function to a simple state reminiscent of the stem cells. An undivided differentiated cell sometimes regains the power of division. This process is called de-differentiation. De-differentiation is a common process in plants during secondary growth and in wound healing mechanism. The plant cells differentiate to become permanent specialized cells of specialized organs. For example, a cell when differentiates to become a component of a sclerenchymatous fiber or xylem tracheids, it loses its protoplasm, develops thick secondary wall made up of lignin and becomes totally permanent. However, some meristematic cells differentiate to become living permanent cells of parenchyma. These cells do not permanently lose their potentiality of division. These permanent cells may regain the capacity of division under certain conditions. So, under natural conditions, the cells of medullary rays or the pericycle may become interfascicular cambium or cork cambium respectively. These are the examples of de-differentiation. Now, here the redifferentiation. What is redifferentiation? Redifferentiation is defined as maturation or differentiation of de-differentiated tissues. So, a de-differentiated cells can divide and produce new cells. New cells produced again lost the power of division and become a part of permanent tissue, which is called redifferentiation. For example, formation of tumor cells. So the cells of cambium becomes meristematic and divide by mitotic divisions. The daughter cells formed from cambium may once again lose the capacity to divide and mature to become cells of secondary xylem, secondary phloem or periderm. This is called redifferentiation. So, a common example of de-differentiation and redifferentiation is the culture of plant tissue. During this technique, a lump of permanent tissues from the portion of pith is dissected out and cultured on the solid culture media. The parenchymatous cells of pith become meristematic and start dividing. This is an example of de-differentiation. The dividing cells form a mass of undifferentiated cells called callus. These cells may be divided to in different manners in order to produce different organs. For example, treatment of certain hormones may lead to the differentiation of shoot buds. Similarly, treatment with rooting hormones may lead to the formation of roots. Thus, the meristematic cells of callus may become permanent tissues by the process of redifferentiation. As mentioned earlier that the growth in plant is open, the process of differentiation is also an open system because the cells or tissues arising out from the same meristem may develop into different structures at maturity. Final fate of an undifferentiated cell to become a mature cell is determined by the location of the particular cell. For example, a dermal cell of developing leaf may become trichomes, guard cells, or epidermal cell. Final differentiation of a meristem cells into a mature cell is achieved by biochemical changes within the cell, but the position and location of the cell plays a very important role in differentiation. So, differentiation is the cells derived from the root apical and the shoot apical meristems and the cambium differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. During differentiation, cells undergo few to major structural changes both in their cell walls and protoplasm. For example, to form a tertiary element, the cells would lose their protoplasm. They also develop very strong elastic lignocellulosic secondary walls to carry water to long distance even under extreme tension. De-differentiation here, after de-differentiation, the living differentiated cells which have lost the capacity to divide can regain the capacity of division under certain conditions. This phenomenon is termed as de-differentiation. For example, formation of meristems, interfascicular cambium and cork cambium from fully differentiated parenchyma cells. And the last is redifferentiation. After the process of dedifferentiation, the produced cells once again lost the capacity to divide but matured to perform specific functions. That is, get redifferentiated. Examples are secondary xylem, secondary phloem, etc. So, what are the difference between the dedifferentiation and redifferentiation? The process by which structures or behaviors specialized for a specific function lose their specialization and become simplified or generalized is called dedifferentiation, while the process by which a group of once differentiated cells return to their original specialized form is called redifferentiation. In dedifferentiation acts as meristematic tissues including interfascicular vascular cambium, cork cambium and und meristem, while in case of redifferentiation serves as the functionally specialized tissue. Dedifferentiation allows the plant body to produce new cells at a particular location, while the redifferentiation here the important to perform a function specific to a particular part of the plant. Example of dedifferentiations are the formation of the interfascicular cambium and the cork cambium from fully differentiated parenchyma cells, while the examples of redifferentiations are the specialization of vascular cambium into secondary xylem and phloem. So root apical, shoot apical meristem and the cambium differentiate and mature to perform specific functions is differentiation. Cell wall and protoplasm undergoes changes. Differentiated tertiary elements cells lose their protoplasm, develop a very strong elastic lignocellulosic secondary cell walls. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions is dedifferentiation. Development of interfascicular cambium and the cork cambium from fully differentiated parenchymatous cells is example of dedifferentiation. Cells produced by the dedifferentiated tissues again lose the capacity to divide and mature to perform a specific function is called redifferentiation. So this is all about the differentiation, dedifferentiation and redifferentiation. After this, now the development in plants. So, development is a process that includes a whole series of changes which an organism goes through during its life cycle. It begins with the cell division in zygote, follows several developmental stages including the formation of embryo, seed germination, formation of roots, stems, leaves, flowers and fruits. Finally, the mature organs undergo senescence and die. The term development is also applied in case of development of specific tissues or specific organs. The process of development is considered as the sum of growth and differentiations controlled by several intrinsic factors, including genetic and hormonal, and extrinsic factors including light, temperature, water, oxygen, nutrients, etc. So development, here the changes in the life cycle. Plasticity, what is plasticity? Different kinds of structure in response to environment or phases of life. Example like heterophyll in cotton and coriander. In these plants, leaves have different shape based on the phases of life cycle as well as the habitat. Development can also be termed as growth plus differentiation. Development is controlled by intrinsic as well as extrinsic factors. Intrinsic like the genetic factors in the plant growth regulators, and the extrinsic like the light, temperature, water, oxygen, etc. So the development of plant organs generally follow a set pattern under a constant environment. However, the pattern may vary with the changes in the environment or different phases of life. That means the morphology of the plant organs follow different pathways in the responses to environment or phases of life. To cope with the changes in the environment, the plant organs also change their structure. This ability is called plasticity. Like heterophyly in case of cotton and coriander and larkspur. So these plants exhibit morphology of leaves well adapted to their environment. The leaves of juvenile plant are quite different in shape from those in the mature plant. So, what is plasticity and heterophyly? Plasticity, the ability of a plant to follow different pathways and produce different structures in responses to environment and phases of life. Like in case of cotton and coriander plant, the leaves of the juvenile plant are different in shape from those in mature plants. Heterophyly is the phenomena of appearance of different forms of leaves on the same plant is called heterophyly, like the heterophyly in cotton, coriander, and larkspur. So, on the other hand, the leaves of buttercup shows the heterophilous development but through different environment, and they are much dissected when grow in aquatic habitats and remains intact when grow in terrestrial habitats. So this phenomena of heterophyly is an example of plasticity. Here you can see that heterophyly in larkspur and buttercup. That is, here you can see that juvenile, adult, and terrestrial habitats. So this is all about the plant development. Here, this is the end of video. Before this, see here the sequences of the developmental process in case of plants. So here this is the sequence of developmental process in case of plant, that is meristematic cells, which protoplasmatic growth, which after the differentiations form the mature cells, and these mature cells by the expansion and maturation converted, then the senescence occur, then death occur, and in the plasmatic growth cell divisions occur. So these are all the sequences of the developmental process in the plant cell. Now, difference between the growth and development. So what are the main differences? Growth is qualitative while the development is quantitative. Growth is objectively observed and measured, while the development it cannot be measured. Growth, it does not continue throughout lifespan, it stops after maturation, while the development it is continuous in all areas of the mental activity. Growth is one aspect of development, while the development is complex and many-sided. Growth occurs in different parts while the development changes in the organism. Growth is not directional while the development is progressive and sequential. Growth is not uniform in all the parts while the rate of development is not uniform in the development. In growth, the individual differences exist while in case of development, children differs in the level of development. Growth is not affected by learning, while the development here, the learning and experiences affects. Growth may or not lead to development, while the development is integrative. Development in one aspect promotes the development in the other aspects. So these are the main differences between the growth and developments, and this is the end of video in which we knew about the differentiation, de-differentiation and redifferentiation and the development in plants. So the next upcoming video will be on the plant growth regulators and the plant hormones. So if you understand this video, please like and share it and subscribe to my channel Rupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.